Hello, today we are going to see some techniques used in cell biology. Some of the techniques mostly used in cell biology are microscopy, subcellular fractionation, cytochemical methods, and cell culture. Microscopy is an imaging technique and an essential tool in life and materials science. Why we use microscopy in cell biology? To study objects, we must interact with them. Generally, the technique of interaction is determined by the size of the objects, and then, when an object is of macroscopic size, and this can be studied by direct contact with this object, but when an object is of microscopic size, such as, for example, cells and organelles, we must study them using agents of similar or smaller size, such as microscope. The microscope is an instrument that magnifies very small objects, for example, cells, and it produces an image in which the objects appear larger. These pictures, produced by the microscope, can also be called micrographs. This means that the microscope helps us to see some pictures that our naked eye can't see. Today there are several different microscopy techniques to improve the visualization of fixed or live cells, such as the optical light microscopy, the electron microscopy, the scanning probe microscopy, and the X-ray microscopy. Adding to this, each technique has subdivisions. The main source of morphometric data on cells has been images from the optical or what we call light microscopy and also from electron microscopy. The cell characteristics analyzed using optical microscopy and electron microscopy are external and internal. For example, some external characteristics that the microscope could give to us are the size and the shape of the cell. And some of the main internal characteristics that we got with microscopy are the shape, the position, and the number of organelles and labeled molecules inside the cells. The subcellular fractionation is a biological technique used to separate different cellular organelles while in the same time preserving their individual functions. How to achieve the fractionation? To achieve the fractionation, we have to use differential centrifugation. In the next, we will know what does it mean. Differential centrifugation is a physical separation of organelles using an instrument called centrifuge. This instrument is shown in this figure. The principle of differential centrifugation is that if we use a sequential increase in speed and time of the centrifuge, it means that if we work gradually on augmenting the time and also the speed of this instrument while the tubes of our cells are inside, we are going to have a sequential separation of organelles in the tubes. This means each time we make increasing the time of rotation and also the speed of centrifuge, this will lead us to separate more organelles. Surely the heavier one will separate firstly. So each time we will take our pellet containing the organelles already separated with a defined time and speed, and we can make the supernatant again in the instruments and change to a more time and more speed to get other separation of other organelles. So we can say that in an example that with lower speed and few time, for example 10 minutes, we can have the heavier organelles in the pellet. This means we can separate the heavier organelles from the other ones. And this, for example, the case of the nucleus. And then if you make a higher speed and we add more time, this will lead us to separate the lighter organelles, for example, the ribosomes. This is another example simplifying the differential separation using the centrifugation. When increasing speed and time of the centrifuge, this leads to separate and to fractionate the different organelles of the cell. For example, when we use in the centrifuge 10 minutes with 800 per gravity, we can have this nucleus in the pellets. If we add more time and more speed, we can have the mitochondria, the lysosomes and peroxisomes in the pellet. If you want to have ribosomes, we can use this one, I mean the supernatant remaining here, and put it in another tube and make it in centrifuge again for 3 hours. And with 200,000 per gravity, we can here have a pellet containing ribosomes. So this figure simplifies the technique. If you have a tissue cells, we can use a technique that is called differential centrifugation, and this to separate the different organelles. So firstly, we make an homogenization of these tissue cells and we have an homogenate. This homogenate contain suspension of broken cells and it contain the components, essentially the organelles. If we make this tube in a centrifuge for 800 G and for 10 minutes, we will have here a pellet and a surnatant. The pellets will contain mainly the nucleus sediments because they are the heavier and we can firstly separate them using this timing in the centrifuge. Then we can take our pellets and work on our nucleus. This supernatant, we can make it in a new tube and we can make it or put it in our centrifuge and use 1500 G for other 10 minutes or 15 minutes. After that, we can mainly have in the pellet the mitochondria, the lysosomes and peroxisomes. These organelles, we can have them in this way and then work on them. Then we can again take our supernatant and make it in another tube and make a centrifuge for another 100,000 G for 60 minutes. It means for 1 hour. After this 1 hour, we can have a pellet and a supernatant. Our pellets will contain mainly fragments of plasma membrane and endoplasmic reticulum. And we can also here take this sediment, take this supernatant, put it in another tube and make a centrifuge for 200,000 G for 3 hours. And after those 3 hours, we can here have our ribosomes that will be sedimenting in our pellets. And it will remain here our supernatant containing the cytosol. So this is simplifying all the technique of differential centrifugation for separation of different organelles of the cell. We just need tubes, tissue cells, homogenate them, separate them gradually using different time and different speed of the centrifuge, and in each time we will have different organelles separating according to their density, and hence the heavier will separate in easy way, that means with less time and less speed, and the lighter will separate at the end when we use more time, going to 3 hours and with more speed. With this, I came to the final of my presentation. I thank you very much for your attention and see you soon. Bye.